Hi, this is Amy Proal with the Polybio podcast, and my guest today is Dr. Resia Pretorius. And she is both a department head and a research professor in the Physiological Sciences Department, Faculty of Science at Stellenbosch University in South Africa. And her team has used super resolution and electron microscopy, together with several other methods, to identify microclots containing trapped inflammatory molecules in the blood of patients with long COVID. And she has recently partnered with several clinical collaborators to trial if HELP apheresis, which is a form of blood filtration, can remove these long COVID microclots and improve patient symptoms. So with that, hi, Resia. Thank you so much for joining.
Hello, Amy. Nice to see you again.
Definitely. So, I guess I could just jump in. You have been studying long COVID plasma now for a while from patients with these, ongoing debilitating symptoms after acute infection with SARS-CoV-2. And tell me what you've been finding.
Over the past few months now, we've been looking at, as you mentioned, the long COVID patients. We also have a South African long COVID registry that people can note their symptoms as well as their comorbidities before they have had acute COVID on this registry. And then also we work with a few clinicians in South Africa and now actually all over the world, also with Dr. Beate Jaeger and other clinicians in various parts of the world to look at clotting pathologies as well as platelet hyperactivation in these individuals. So we've looked at a few hundred samples over the past few months, and it is very clear to us that all of the patients, if they are diagnosed correctly, and that is very important to note, that they should be diagnosed with long COVID by a clinician, where there's also a very clear distinction between other possible diseases that they might have, which might mimic long COVID symptoms. But if we look at the patients that are definitely identified as having long COVID and fit the criteria, we have noted microclotting and hyperactivation in all of these patients.
Resia, okay, that's really interesting. And now when it comes to these microclots, you use special microscopy, so microscope work to find them, correct? So does that mean that other teams or other people can or cannot find them? Is it something you can do only? How does this work?
The microscopy method that we use is actually quite easy to do if you've got access to even just a normal light microscope or a student microscope. So we use in our laboratory, we use a fluorescence microscope, which is obviously a quite an expensive piece of equipment. And we use the fluorescence simply as it's an easier method for us to identify the microclots, but we also look at platelet hyperactivation. So the process for that is quite simple. You just take a citrated blood tube that is drawn at room temperature, and you spin down platelet poor plasma, and to that you add a marker that's called theoflavin T. Now, theoflavin T is a well-known marker for amyloid protein, and it was previously used to identify misfolded protein in brains of Alzheimer's patients. So we discovered it a few years back together with my very good friend and collaborator, Professor Douglas Kell, that it actually also binds to misfolded fibrinogen that causes microclotting. So we just use that, very cheap, and we add it to the platelet poor plasma. We put 3 microliters of plasma onto a microscope slide, and that we just view. And with that, then you could see the microclots. You could also just simply do the same. You don't even need to add the theoflavin T. You could just create platelet poor plasma and view a smear like a normal blood smear, but just not as wide over the whole microscope slide. Just try to keep it quite compacted, the little area that you want to look at. You could just do that and look under a normal student light microscope. In fact, one of the long haulers from the USA contacted me a few weeks ago, and she could actually just use this method with a small little one-piece eyepiece microscope that she then took a micrograph or a photo with her iPhone through the microscope piece.
Wow.
Eyepiece. So obviously it's not, it's very crude. You can't, but you can at least see the clots. So it's not publishable, I think, type of micrographs, but at least you could see the microclots. Then for the platelets, it's a little bit more complicated. After we spin down the plasma, we use the hematocrit, which is the red blood cells and the white blood cells and the platelets all trapped at the bottom of the tube that you now spun down. And we also just take that, add 2 markers. One is a marker, a fluorescent marker or antibody for platelets themselves, and the other is for P-selectin, which is a well-known platelet activation marker. We just add those 2, and then we can look at platelet hyperactivation. The reason why we use the hematocrit is that during the process of the spinning, the platelets are very active, easily activated. Now, in a healthy individual, platelets are supposed to be small round balls. They do not activate very much with the spinning, although they do a little bit. They will put out little pseudopodia or feet-like extensions. But in the case of a long COVID blood sample, the actual platelets are so fragile that if you just put them onto a microscope slide, you could just see them massively activated just by this simple spinning process. So it's actually more like a test for platelet fragility that we look at. So this is also quite easy to do, but perhaps not as easy as just looking at the microclots.
Got it. Wow. No, it's really cool to hear you walk through the various steps that you do and just understand the way that your lab works on this. So basically the way I see it is, your lab has the equipment set up and honestly experience, because you've been working on blood coagulation and clotting for a decade, including in other conditions. So you can visualize these clots beautifully with your current microscopy and also the associated inflammatory molecules, the hyperactivated platelets. But it's possible that you can, from that work, create sort of a knowledge base in which other people and other teams or even clinical centers might be able to find these clots in similar ways with related microscopy. So in other words, eventually this could spread so that someone who has long COVID in another city or place could also be examined for these clots?
Precisely, that is what we hope. That it is such an easy method that many people with some sort of microscopy equipment can use it. We are also working with a few international global researchers all over the globe to actually see if we could not translate the microscopy work to flow cytometry, so that the high throughput, because it also works with fluorescence. And THT could possibly be used in a flow cytometry setup, which will obviously be a good idea if one could speed this up. Now, just about the microclots and why it has been missed, or many people feel that they go to a clinician and they are misdiagnosed. And this is one of the most important considerations, is that many of the typical laboratory blood type analysis that you will do at the path lab will not pick up any differences in inflammatory markers, and the patients become very desperate as it's then ascribed, or the condition is ascribed to a psychological issue. And the issue is that if we don't have markers, or we don't have methods that are in pathology laboratories, it doesn't mean that the disease doesn't exist. It means that we have just not found the correct markers. And we do believe that now we have found them. And we recently also published a paper to actually show that the reason why people are not finding, with normal pathology, regular pathology, ELISA tests, any of these increased markers or inflammatory markers or whatever they're looking for, is because the inflammatory markers are actually trapped inside these massive clots. And these clots are really, really big. And as you mentioned, Amy, you will be putting up some photos, micrographs from our lab. And they are really extensive microclotting. And when we did our recent paper that was published in Cardiovascular Diabetology, we explained that we struggled to break the clots or to disentangle them or digest them with a trypsin digestion protocol. We only could digest the clots after a second try. And then we found all of these inflammatory markers inside the clots, including molecules that prevent the clot from breaking up. And one of them is alpha-2 antiplasmin, which is a well-known molecule that it prevents lysis. So your body do not actually have the capability to break down the clots because these molecules prevent it from breaking it down. So yes, the microscopy is just a research tool, but hopefully from there, we will be able to proceed for more mainstream pathology tests.
Yeah, that's very exciting. I mean, and so, you know, with these clots, like you're saying, they're big, they're difficult to break up. So, okay, when I tell people about this, because this is exciting work and I tell lots of people and teams, they say, how does that not end up killing the patient actually? So how do you think that, you know, you have these big blood clots, but are they flexible? Are they able to get through vessels in a way that doesn't result in just a blunt stroke? Or what do you think is going on with how the clots, you know, people, I'll just be honest, stay alive with these clots?
There are a few answers to that. And the first is, if we compare the microclots just visually by looking at the clots in acute COVID versus long COVID, in acute COVID, the clots does seem to have a more opaque consistency, where in long COVID patient, it seems as if they are more spongy, that they might be able to squeeze through fine blood vessels. And the issue with the microclots and the platelet hyperactivation, it all is driven by and drives endothelial damage. Now the question is why don't we see more events? We probably do, but it's not reported, and that is an important consideration that we must look at, because there are probably people dying of these microclots and having long COVID and not being diagnosed and not even know that they've had microclots causing the resulting dilemmas. So the issue with the microclots is, and it comes back to the more than 150 symptoms that have been identified by various organizations like the Long COVID Association in the USA and many published papers. There are over 150 possible and many more symptoms. And if you look at the symptoms closely, all comes back to a hypoxia of certain organ system, whether it's the muscle not getting enough oxygen, whether it's liver damage, whether it's brain fog, concentration issues. One can all bring it back to a reason why the symptoms might happen, because of oxygen deprivation to certain areas. And why does people get some symptoms and not the others? It can be simply something like genetic predisposition or previous consideration of their comorbidities. So we see here from our South African registry that the main symptoms that are reported by most of the long COVID individuals is the fatigue, the massive fatigue and brain fog and sudden low oxygen levels. And that one could all basically, I think, eventually trace back to some sort of oxygen hypoxia or oxygen deprivation of specific organs. And then obviously also many people complain of the muscle weakness and they can't walk a few steps or climb stairs. So I think we are missing many of the events due to misdiagnosis of the long COVID patients.
I understand that. And what you mean then is that someone could have gotten COVID and then actually had a cardiac or stroke type event, and we almost wouldn't have ever checked if that patient had microclotting in their blood. And so we are looking at people who, you know, did survive more or whatever, but it doesn't mean that some people didn't even have an acute event, which is, yeah, something that obviously has to be studied urgently. I do think then with the clotting and the stuff that you're already identifying, so and just to review, what you think is happening is that the clotting and the inflammation associated with the clotting damages the blood vessels, so the vasculature, and then that prevents the blood vessels from being able to distribute, because usually blood carries oxygen with it. It prevents the blood vessels from able to carry oxygen to a broad range of tissues in the body. And so then you lead to a what's called hypoxic or low oxygen state. That's the idea. Yeah.
Yeah, that and also the microclots just simply block the vasculature, so that the red blood cells cannot get to the areas to have the oxygen exchange and the CO2 exchange at the end areas. And also with this constant circular inflammatory process happening, the microclots drive damage to the endothelial layers. The endothelial layers then produce more von Willebrand factor and tissue factor, and that just drives inflammation all the time. So we need to somehow address that, so that the endothelial layers, the microclotting, and the platelet hyperactivation are all addressed, so that there's a general healing of the vasculature and the whole blood flow system, I would think.
Yeah, absolutely. And then in terms of the way that the clots form in the first place, what do you think occurs? So if someone, you know, is infected with SARS-CoV-2, and then how do they end up with these clots? Could the spike protein be involved? What other aspects of the initial disease do you think contribute to the formation?
So during acute COVID, it's now well accepted, and perhaps it was less well accepted a few months ago. It was initially thought that it is simply a respiratory and a pneumonial disease right in the beginning. But now I think more people have come under the impression, with more research coming out of various parts of the world, also from our laboratory quite early last year already, that it's actually, although a respiratory disease, as we know it, affecting the lungs, it is mostly also affecting the vascular system. And driving this whole process during acute COVID is then the spike protein. And the clotting happens during acute COVID. So also, all the patients that I have seen with acute COVID that has come via the ICU and my clinical collaborator under ethics, obviously, all of them had severe microclotting and platelet hyperactivation during the acute COVID phase. So I think if the issue is, if the person then is not infective anymore, whether the person has been treated in ICU or just simply at home with oxygen or not oxygen, or even some people being asymptomatic but testing positive. We have had many cases like that reported all over the globe, that people didn't even have acute COVID so badly, and then they have these long COVID symptoms. So the issue, I think, is that in the acute phase, the clotting and the platelet hyperactivation is not addressed fully. And then the patient just continues, not being infective anymore, but just continues with clotting, vasculature damage, platelet hyperactivation that is not resolved properly. So acute COVID needs to be, I think, addressed. The clotting needs to be addressed in acute COVID, so that you don't end up with people sitting with long COVID issues. And as you mentioned rightly, there can be many factors, but many inflammatory molecules are dysregulated during acute COVID. But we have also shown that the spike protein, when we buy the spike protein from Sigma or Merck or one of those companies, and we add it to healthy blood, that we can actually trigger both platelet hyperactivation and microclot formation in the laboratory. So we do think that spike protein plays a crucial role in the initial activation of the amyloid fibrinogen molecules, and then it just like a domino effect, just perpetuates the microclot formation, and we need to prevent microclotting, I think, so that the body can then get rid of the microclots that are in circulation and then heal itself that way. So definitely spike protein, but also other possible viral, bacterial molecules that might have been in circulation or persistence of these bacteria and viruses, probably or might be from previous diseases. We still need to figure that out. So we don't actually know that pathology quite yet, but I think many groups are looking into that. And I think very needed research.
Definitely. No, I mean, it's very interesting. It seems like the spike protein has unique properties to drive the specific kind of these, larger, difficult to digest microclots you're seeing in long COVID, which seems, because you've done previous work, because I've known your work for many years now, where you've shown in other conditions, Alzheimer's disease, Parkinson's disease, that there can be a degree of clotting in the blood of those patients as well. And that may also be from contributions of pathogens or other inflammatory issues that are going on in those patients, right? But those are different. The clotting is somewhat different than what you're seeing here. So it seems to me like the spike is able to catalyze sort of a unique form of clot that seems sort of somewhat long COVID unique, which is interesting because the spike protein is an interesting protein, to be honest, that's still being studied. And I do think, I think it's important to explain, you've done work with platelets, in which you show how platelets have receptors that can recognize proteins created by a broad range of different pathogens. So do you think that that's where you're coming in with the possibility that, let's say you had SARS, you got COVID and Epstein-Barr virus happened to reactivate, which is something that's being documented in acute COVID, then platelets actually have receptors for Epstein-Barr protein, correct?
That is correct.
What other receptors, what other pathogens do platelets recognize? A range of bacterial, viral, many?
A range of bacterial, viral, the membrane parts of the virus and the spike-like proteins of other viruses, not spike protein, obviously, but even HIV and those type of the molecules, inflammatory molecules from those viruses and bacteria. Platelets can recognize. It can bind to the membranes. It can activate the cascade inside. So from outside in cascade activation, which then result in an inside out cascade activation, where the platelet just hyperactivate and just dumps all of its content into the surrounding circulation. And that again can activate the receptors on red blood cells, on all of the other cellular, the monocytes, all of the other white blood cells. They form complexes with platelets, and then they can all bind to the endothelial receptors. So it's just one massive activation happening via spike protein, other viruses, LPS from the walls of certain bacteria. And you correctly say we have looked at the platelet activation and also how fibrinogen misfold in the presence of something like LPS from bacterial wall components. And that also drives both platelet hyperactivation and protein fibrinogen plasma protein misfolding. But I must say the extent that spike protein does this is just mind-boggling. It's just absolutely, I've never seen the amount of the size, the sheer size of the clots and the amount of clots in individuals with acute COVID and long COVID. Obviously, in people with inflammatory conditions, you do find hypercoagulability. I always say that one of the important hallmarks of inflammation or all systemic inflammatory conditions will be a pathological clotting profile. So there are pathological clotting happening in type 2 diabetes, psoriasis, rheumatoid arthritis, you name it. If there's inflammation in the systems, in circulation, you will find hyperactivation of platelets, fibrinogen, and to a certain extent also endothelial damage. But the sheer volume and size of the dysfunction. I've never seen it. And obviously, if you have got high blood pressure, you've got diabetes as a comorbidity before you develop acute COVID, then we could just imagine the person's system is already hyperactivated. It's already damaged. So it's just then why we also see these individuals with having a poorer outcome of acute COVID, and many of them struggling with long COVID.
Absolutely. That's one of the things that, I think about is, you have patients who get COVID, and not everyone goes on to get long COVID and have these coagulatory issues. So some of the reasons that certain people may develop the clotting more than others may exactly be either partly their human genetics, in which there are human genetic variants that already kind of predispose towards increased clotting. If your human genome might already be, lean in that direction. But then also, if you already, because platelets can respond to a broad range of viral and bacterial pathogens, if you have ongoing microbiome issues, and so there's more bacteria already leaking into your gut, leaky gut, or problems from the mouth or whatever, you're going to have more organism in the blood already that, let's say, the spike protein starts to catalyze this clotting. The other organisms or other underlying things that can additionally activate platelets can join in, and those people might have a worse case or more difficulty or increased clotting. So that's almost like there's these predisposition in sense of like human and organism based predispositions to why certain people may develop more clots than others, I guess.
Absolutely. And if I can also just mention, I've got a very good friend of mine that's also, so we're always looking for good controls. So this friend of mine in her early 50s developed acute COVID, and she didn't even realize that she was sick. She actually went for a random PCR test because she needed to go on a weekend away, and she needed to do the test, and then she discovered she had acute COVID. So she was an excellent case for us to look at, someone who we could get blood from every day, and follow her through her acute COVID phase. And she also had severe microclots, and she didn't even know she had acute COVID, and obviously were tested. And we looked at her, and she became ill. She was not as if nothing, but she, what was interesting, after she progressed through the disease, and perhaps 2 or 3 weeks after she wasn't infective anymore, she still had the microclots, some of it. She still had some platelet hyperactivation, but then over the next few weeks, it just disappeared, and she became totally healthy again. So in her case, her fibrinolytic system, all her pathological pathways were activated, but then her body could take care of it, and she could return to normal clotting pathology, or clotting normal physiology, actually, not pathology. She could return to that by just simply her own body's reaction, because she's a fit person. She doesn't have comorbidities. She, as I mentioned, regularly exercises. She eats well. She's not overweight. She doesn't have anything that predisposes her to a significant clotting hypercoagulability pathology in the first place. So it's that was quite interesting for me to note that she also had all of these clotting dilemmas, but then she returned to health after a few weeks.
It is a truly interesting case, because first, it's just interesting that you don't have to have a serious case of COVID to develop these clots. That's interesting in and of itself, right? That you can have a mild case and still develop clotting. And then second, it really does emphasize then that, one thing that will be interesting to study will be, what underlying factors patients have that may, prevent their immune system from sort of clearing up the clots on it. The immune system may have an inherent capacity to clear these clots up, but let's say it's already, dysregulated because of another Epstein-Barr infection or something else, or other inflammatory issues. Perhaps it's held down and it can't do it, right? So that might be, you might have patients who, understanding those underlying issues may also be part of the long-term vision for how to really, figure out who's going to get more clotting, and also how to get them fully better, which would be the goal, right? That's really interesting. So, and I guess one thing that would be possible, I'm not sure, is that if some people don't fully clear SARS-CoV-2 over time, which is demonstrated in some studies, that there could be sort of ongoing release of spike protein that could probably aggravate the clotting for a very long period of time. So I assume if that was the case.
Absolutely. I think that might definitely be the case. And then it just also again shows the significance of the spike protein, and that if you don't have anything else going on, the spike protein anyway causes these microclot formation and damage to your endothelial layers and your platelets. But somehow your body and your immune function, when it's working well and it's functioning well, can get rid of it and you don't have an issue. But the issue is, if we look at the amount of individuals that simply do not get better from acute COVID and go into long COVID, it tells us that there are pathologies, underlying pathologies that people might not even know they have, or as you mentioned, genetics or gut dysbiosis, or it would be interesting to correlate all of those comorbidities and ongoing things in a very large study. I think that is definitely needed, and we need to urgently look into that.
I agree. There's a couple of teams that I'm curious to bring in on some of that for interest, where we could do more of the, microbial content, microbial viral content of the clots, of the blood itself, the, protein, just for interest, because who knows, it could be interesting to understand any of the factors that could be contributing to this. So yeah, that is a cool long-term thing. So then, okay, so you have now been able to help actually remove these clots from patients. So, then the question becomes, what do you do when patients have these microclots? So you've been collaborating with some clinicians using what's called apheresis to remove the clots. So will you explain that more?
I work with two teams. So in South Africa, I work with Dr. Jaco Loubser, and he uses, in the long COVID cohort, he uses a method where he treats the clotting, to prevent clotting from continuing. And he also prevents platelet hyperactivation, but that is more like a pharmaceutical intervention. And I must say very clearly here that this can be very, very dangerous. So please don't just go out and go and get anticoagulants, because if you're prone to bleeding, you could die. So it's a really under good supervision of a clinician, making sure that you indeed do have microclots, that you do have platelet hyperactivation, and a clear clinical investigation to confirm that you actually are hypercoagulable with these microclots. So that is a pharmaceutical intervention, and currently it is still seen as an experimental protocol. And we are working with a lot of clinicians all over the world to try to see if we could possibly have a system where we can have a proper trial to investigate what can happen. But then very interestingly for us is that we worked with Dr. Beate Jaeger and a whole group of clinicians from all over the world using HELP apheresis. HELP apheresis system is in Germany in Dr. Jaeger's clinic, where she filters out microclots and all of the other, it also filters out fatty deposits, and it might filter out autoantibodies or inflammatory molecules, where it actually removes the clot load. And we were in Mulheim at her clinic with a whole team of South African investigators that traveled with me from my blood lab, and also a whole team of clinical clinicians helping to look at before and after treatment. And that really seems to be working. Dr. Jaeger's actually had a whole series of patients that she treated with HELP apheresis very successfully, where they then really become healthy again.
Wow. Yes. And now I've been on some of the meetings over that, and it's incredibly exciting. And it's very cool to actually even hear from some of the patients themselves describe that they feel better. And you've characterized the clotting before and after the apheresis, correct? And that's something you're going to publish soon? And
We
you
did.
obviously, it's not probably giving too much away to say that there's less clots after the apheresis.
Absolutely.
Yeah.
And we are very, very excited to also report that we were working together with Dr. Martin Crater, and he is from the Max Planck Institute in the adjacent city from Mulheim. He was also in our laboratory. And what was so exciting for us was that he had a microfluidic system. Sorry, here's a fly here in my office. He actually had a microfluidic system where he looked at the red blood cells and how they travel in his microfluidic system. And we actually are in the process of comparing his very exciting results with our static microscopy results. And yes, we can report that there was definitely a significant reduction of microclot presence after the apheresis system. And we looked at 18 individuals, naive, not been treated previously with HELP apheresis. We looked at before they had their treatment, 6 hours after, and then 24 hours after, and we're comparing the various time slots. We also did quite a very complicated clinical workup of the patients, and there it was with the help of Dr. Asad Khan, which we all know now. He's been talking about his disease. He also has got LongCovid and is actually himself very sick, but he did a wonderful job where he looked at the patients, did their walk tests and spirometry, and they busy analyzing that as well. And he also worked with Professor or Dr. David Petrino from the USA, where they also looked at various different factors that might affect the person's ability to function. So we're very excited to say that we are currently preparing the data, and the whole international team, and we're very thankful, Amy, that you're also part of this team. We all will put out a paper with everyone as co-authors, and we hope to have a draft ready very soon, and our results are really good. Now, obviously, we only followed the patients while I was in Mulheim for the second day or the day after, after apheresis. And the plan is, I hope that it would be able to realize that Dr. Asad Khan and Dr. Beate Jaeger would be able to follow up those 18 patients in 9 weeks or 12 weeks after they had the apheresis. Dr. Jaeger also put those 18 patients on a pharmaceutical regime of Dr. Jaco Loubser, because we're all working together. So they are also treating some of those patients, I think all of them actually, but some of them might stop or have some other regimes added depending on how they feel. They are on treatment using a protocol where they calm down the platelets and prevent more microclot formation. But more on that, I hope, in about 9 weeks after, or 3 months after the treatment. We'll definitely need to do a follow-up, because obviously this is such a debilitating disease, and one apheresis treatment is probably not going to be enough.
Yeah, that makes sense. It's very cool that you already found a change in clotting with one treatment, but I agree with you. There's you continue to look at the long-term health of the patient, how it works over time. Obviously, it makes sense. I have to say that it's interesting with the microclots themselves. It seems like a liability for the patient when they're sick, that the inflammatory molecules, potentially viral or bacterial proteins that might be involved, autoantibodies or anything like that are trapped in the clots. That is actually probably not good for you when you're sick, because it's harder for your immune system to find them or target them if they're hidden in a clot, in a simple sense. But that's a pro for the apheresis you're describing, because if you're able to remove the clot, you're able to also remove potentially the viral bacterial proteins, inflammatory molecules, autoantibodies that are also actually potentially driving this. So that seems like a real added benefit, that you may be removing quite a number of pathological issues along with the clots, which is really cool. And then with that, I think this is an open question. There are centers that do apheresis, right? Here in the USA and other places. How much do we think that the HELP apheresis and that technique of doing apheresis can be applied to what I will call normal, other apheresis that is more available at other centers now?
That is a question that we definitely need to look into, and I do believe Professor Moriarty from the USA, that's also in our team, is actually looking into that, and many other groups are looking if the same effects will be in patients treated with, as you mentioned, the normal type of apheresis equipment. I do know we don't have the HELP apheresis system in South Africa yet, but I do know Dr. Jaco Loubser have treated one or two of his very ill acute COVID patients with the more the type of apheresis machines that we have here. But he's very hesitant to use the machines that we have in South Africa for long COVID, because it's not as easy on the body as HELP apheresis. So I do think Professor Moriarty and the rest of the individuals working on the other types of apheresis machines, not necessarily the Braun machine that was in Mulheim and in the clinic of Dr. Beate Jaeger. It's definitely, I think, important to see what the effects of those other types of machines are, also in removing microclots and calming down the whole inflammatory system. I do believe that the other types of machines do remove some of the inflammatory molecules, and some of the autoantibodies might be removed. So definitely there's some interesting research that need to be done in a comparative nature.
Definitely. No, I think that's one of the next steps is other trials involving, different forms of apheresis to see how much that the clotting seems to diminish and whatnot compared to the HELP apheresis that you're already using, because it's amazing to think that there could be a growing trend in which different long COVID treatment centers offered apheresis. It would just be, obviously, we need to know which machines are able to do what and what with the clot, which is an ongoing, really cool research consideration. And then also with the drugs that, for example, Jaco is using for the platelet, then I understand that you really need to only use these if you already have a doctor who's characterized clotting and whatever and can administer them in a specific fashion. But there are existing drugs then that can calm down platelet activity and can, to a point, help with the decoagulation.
Absolutely, they are, and basically we need to give patients drugs that calm down the platelets, but also simultaneously work on the enzymatic pathway to prevent new clots from forming. Okay. Now, for that, we urgently need a proper trial. That is the only way that we will be able to show with confidence that we're not pushing people into bleeding, and that they are correctly identified, the groups that might receive this type of treatment. And we are also talking to various parties that might be interested in embarking on such a trial. So I really hope that this would be in a year time from now, we would sit here again and talk about the trial outcomes. It is the only way how we're going to solve the question, because now there are all sorts of people on, long haulers themselves, doctors, researchers on Twitter and in papers saying that we must do something or this or that, but we can't do anything really and suggest this is the regime if there is not a controlled trial. And this is the first and most important thing. But before the trial, we need to establish the proper diagnostic pathways. So the diagnostic should be based on identifying microclots and platelet hyperactivation, and then many other inflammatory molecules that you would want to look into: alpha-2 antiplasmin, fibrinogen levels, von Willebrand factor, all of those molecules, perhaps some autoantibodies that might be identified. we must have a proper, well thought through process and clinical trial setup that we compare individuals perhaps in a staggered way, where some will be on anticoagulation, some on help apheresis, some on other types of apheresis, and then swap around the patient group so that everyone can have the benefit eventually, but in staggered way. We know that there have been a few publications that show that there is not good outcomes or not significant changes when, for example, Clexane is used. But the issue is, when we look at those type of trials, the trial did not focus on blocking the enzymatic pathway as well as the platelet hyperactivation. So if you don't do a two-pronged or multi-pronged approach to calm down platelets and prevent microclot formation, you will always fail, because if you just treat the one, the other perpetuates the disease. So it's just one big circular mess of inflammatory hyperactivation. So one needs to go in and do the trial with various multi-pronged approaches to find out what is the best scenario, and perhaps there's other medications as well that can use, not only the ones typically used for calming platelets and for preventing microclot formation, but that desperately needs to be done. And we cannot stress that enough.
It does. No, I agree very much, because that, I have patients asking me, and what could I take for anticoagulation? And that's the thing, is there are some medications. That's a wonderful thing. There are existing medications that may do this, but exactly, without a formal trial to evaluate when to use them, how to use them, what are the best outcomes, we're going to end up with a mishmash of hard to understand outcomes. And so yeah, I agree with you that so basically a randomized controlled trial that you could start in South Africa at Stellenbosch, you would be able to start the trial if you need funding?
Yes, I think so. We do need funding, and we could do it here in South Africa, but it would be also great if you could do it in other parts of the world. We have been talking also to Professor Ashley Woodcock from Manchester, and we're thinking that it might be an idea to perhaps do something in the UK, and even if we do have funding in the USA. But as you mentioned, it is massively expensive, and currently our research lab is working on a shoestring budget. We could hardly do our microscopy work currently, because funding is just so constrained in South Africa. So if there's funding and a proper entity that does do this as a mainstream job, what they do, we rather need something like that, where it's a reputable group of individuals that run a randomized trial in a well coordinated system. So although we would love to do it in South Africa, I don't think we have the capacity, but we could definitely be involved in some ways.
Yeah, that sounds reasonable. I understand it's money, but it does seem like something important to do right now, especially when you're explaining that, even patients who don't develop long COVID, as a defined condition, seem to even develop these clots. It seems like something that is impacting a large number of people in the world right now, as we continue to be exposed to different variants of this virus, which actually is a question of mine. Do you think that the different variants of SARS-CoV-2, do you think that the spike differences, do you have any data on that? Do you think that, I don't know how you could have anything with Omicron, but for example, is Delta, do you think, have you been able to tell if the Delta spike or anything seems to do anything different with the clotting, or is there a change with that?
So in the patients themselves, we didn't see, if the patients were very ill, they had clotting, irrespective of which of the variants they had. We did actually buy the spike protein from the different variants and looked at how they affect, not from Omicron yet, but from Delta and the previous ones. And we couldn't we couldn't identify differences in what they do to the blood. So definitely they do both, and also we did have spike protein from the South African and the UK versions of, and we couldn't see differences in clotting when we added it to the blood sample, but also then we was looking at patients from the first wave to right through to the fourth wave in South Africa. If they were sick and they had complaints of shortness of breath and all of the symptoms of acute COVID in the ICUs and which Dr. Yaku treated, and in some of the other clinicians that we work with, if they had the symptoms, the clotting was there and the platelet hyperactivation was there, as seen with our microscopy methods. So now the question about the other variants, that is an open question. So currently in South Africa with a new variant, it seems as if the ICUs are not really overtaxed currently. So in fact, I have not seen patients' blood sample with a new variant yet. But that does not mean that we will not see them in January, when over the Christmas holidays, when South Africa just shuts down completely summer holidays and everyone goes to the beach and have parties. So we might have a influx in January into the hospitals. Then we might see the effects on the blood. But what I'm more worried about is actually the fact that although the people are not necessarily getting so sick, the question is, will there be a population that weren't very sick, that perhaps, again, was asymptomatic, but then sit with this long-term, long COVID type symptoms? We simply don't know.
I agree with you. It's one of these things where, for acute COVID, you say, okay, it's mild, that's a good thing. But then when we now realize that mild cases of COVID can still result in the microclots, in long COVID symptoms, and really debilitating disease, the fact that a certain variant might be more mild isn't even that comforting when it comes to this chronic sequelae that can happen from this, And, I personally worry a little bit that with the Omicron variant, evading the immune response so much, it might be even more difficult to clear this strain, for people to fully get rid of it, because it, evades the immune response. So you could have more spike protein over time. It's like not ideal. So another reason to continue to do your work in trials and research at a very accelerated pace. So then I guess the goal is to just recap. So, because you have a lot of things going on. And to be honest, I've been so impressed at how fast you have worked in the last months, and how many collaborations and how many things you have actually gotten to move forward. So on one hand, you're perfecting the diagnostic capacity of this. That's some work done in South Africa to perfect the way that you can sort of identify clots and what proteins and stuff are going to be part of sort of the kind of, I'll say, the key diagnostic that you increasingly use in trials, in other randomized control issues, to identify the clots. But then you also have the aspect of treatment. And in that sense, you are seeking support to do a trial of both help apheresis more formally. You have one already existing study, but you could do a larger number of subjects. You could do more with the apheresis outcomes, and also then a randomized clinical trial of anticoagulant, drug type or platelet inhibiting drugs that could be used in these patients as well. So those would be your main goals moving forward then?
Absolutely, that would be the main goals. And just to state that the study that we did in Mulheim was not a trial. It was basically just a study of tracking Dr. Beata's patients that she already was seeing through the various treatments. So for it to be a trial, we would have had to have it registered as a trial at an ethics committee. So we didn't have that. We just traced the people, or tracked the people through their normal best clinical practice processes. But saying that information will definitely have much information to be presented as a case study for the trial. So definitely exactly. that is what we need. And we also desperately need to look into what is happening with children. Yes. Because children and long COVID is really understudied and underreported, and not seen as a key fact of disease. And if we don't treat the children with long COVID, or at least see how we can diagnose them, we're going to sit with a population of adults in a few years' time that are really useless for the economy.
Yeah.
And their health would have deteriorated so much that really individuals that could have made a significant impact on the economy and their households and becoming someone and going to university, they're not going to have that. And it is so heartbreaking to know that we're going to lose millions potentially of individuals that will be permanently sitting with disability. And one can just think what the economic impact it will have on households, but on the economy of countries if we lose a whole population of individuals that were young people in this 2020 till 2022, 2023 timeframe. So we urge you to look at.
I agree. It's an emergency. It's incredibly pressing. And, what also makes it extra pressing is that it's somewhat anecdotal, but your data shows that the earlier you treat, the earlier you try to address these coagulatory issues, the earlier you try to intervene with the clotting, even right away in acute disease, the less likely you are to develop chronic problems with it. But once it becomes chronic, it's able to self-perpetuate in lots of ways. If the virus doesn't clear, the spike protein could keep augmenting it, or other organism-based type stuff, or just inflammation or ongoing issues. So the more that you don't address this, the more it can become more and more of a problem. The more and more of the blood vessels in a patient can become damaged, the more that will have, and that becomes a huge process to reverse and repair. So it just seems really important that this research is done soon also, so that earlier intervention can be something that people do before they become very, very sick for a long period of time. So yeah, I agree, it's very important. And then finally, we're also trying to I'm trying to work with you, Tinge, here to move this work into ME/CFS as well. So, related diagnosis in which patients usually develop debilitating chronic symptoms, but after other infections with not SARS-CoV-2, but herpes viruses or enteroviruses, a majority of cases. And so that's something that we're, trying to get going in this January, but already you've recruited some patients through a very helpful South African organization. And so that seems, you want to talk about that quickly.
We are very excited to work with you, Amy, and with also using mic system with identifying patients properly, and with the wonderful team of Resia Pretorius from the South African Association, the ME/CFS Association here in South Africa. She's given us access to patients. We've got a group of patients of about 60 that we identified, which we already received informed consent from, and we've already completed Mike van Elzakke's grading system to make sure that they do have the correct criteria, that they do have the disease that we are looking for. And we have got a fantastic PhD student, Massimo Nunes, who was a student of mine that I've been recruiting these patients, and we do have ethics for that. And we are now ready to go in January when everything opens up again to collect the blood samples from these patients. And the plan is to look at their blood, to look at platelet hyperactivation, to see what is happening with microclots. Perhaps we won't see the same profiles as with the long COVID. Perhaps we'll see similar things, but we will not walk this in front of it. Let us look at what we see. That is the most important thing. And then we also plan to do proteomics. So that is very important. We need to determine which molecules drive this very debilitating condition. It's been a condition that has been very much understudied, as all of your listeners will know and will report on. And it's just a very, very understudied, under-researched area, and unfortunately also by many being seen as a psychological issue, and which it is not. Once again, if there's not a marker, it's not to say that there's not disease. We just haven't found the marker yet, and we need to do that urgently. So we're very excited. We are waiting for the outcome of a grant. We hope to have an outcome by middle of January. If we do get that grant, we could do everything that we want to, because the grant will be enough to do proteomics and do lots of other things. So we're just really hoping that the grant will come off. But if we don't, we do have had some wonderful sponsors from some of the community, from a family that gave us some funding already. And we will just then have to raise funds for this study. We could do a lot only with microscopy on a shoestring budget, but that we don't want to do that. We need to find the molecules, and for that we need expensive proteomics. So we will do our best, and we will collect all of the samples in January. And we have just also developed a new method to enzymatically, but also chemically digest the microclots. We developed the method with our 100 LongCovid patients that we could study and collect about 4 or 5 months ago. We're nearly ready with that on a bigger cohort. So we do have a quicker method to digest the clots. So I think the process to do proteomics would be a little bit quicker than the LongCovid study. And we hope if we could run the proteomics end of January, we will have results 2 months later. It sounds fast, but proteomics is really complicated. Yeah. It's not a simple point of care test. It's very complicated, and you must choose your molecules that you search for carefully. But we will do that together with you and some of the renowned researchers in the field. We will try to find and search for target specific molecules that we search for to see if there's could find viral, bacterial, and then all of the other molecules that have been previously noted to have an effect. We'll go in search of them.
Yeah, I'm excited. I agree. It's very cool. Looking forward to January, where hopefully we get the grant. If not, we'll make it happen. That's how I feel. So we'll work on that. But yeah, because it's great because, honestly, I, approached you about studying coagulatory issues related to viral and bacterial proteins in ME/CFS before you actually started your long COVID work. And that was something we were already discussing. And I interviewed you once on that once before. And then it's kind of funny because the order switched where COVID hit, and don't blame you. You started identifying these totally unique microclots in long COVID. But now it's cool to kind of circle back and be like, yeah, let's go back to ME/CFS as well, where, there's just existing potential even before the long COVID work came out to better understand the possible coagulatory issues in those patients. So yeah, I'm definitely excited for that work. And, last one thing, it's just a thought, but, patients with ME/CFS and with long COVID now are increasingly diagnosed with what's called small fiber neuropathy, where the peripheral nerves of their, especially sometimes in the legs, will die off. And then they have, problems with the nervous system. And I do think it's interesting to think about the possibility that maybe the blood vessel feeding these small nerves could die off, which it might, because the blood is coagulated along the lines of what you're finding. And so it's too thick to really get into the tiny blood vessels that feed the little nerves that are involved in small fiber neuropathy. And so I actually think that there's a connection between the coagulatory issues that you're finding and the small fiber neuropathy, which is another topic that some of the teams we work with are studying in these same patients. So I think there's some other cool connections that we can make on those topics as well. So there's just, what I like about your work is that it brings together a lot of important themes that people are working on in these communities and starts to unite them more, in that you have the bacterial and sometimes viral generally, but even sometimes bacterial organisms that these patients can harbor. Those pathogens make proteins that can directly activate platelets, which could be part of a picture of this. But there's also other inflammatory issues and cytokine issues that other teams have noted that can be also exacerbate coagulatory issues. And then, like you said, once the blood vessels become damaged and the vasculature and blood isn't able to circulate well throughout the body and you're losing oxygen, you can get into some of the other symptoms that people start to experience. And then you can even start to think of something like small fiber neuropathy, or how the nervous system isn't getting the blood that it needs. So you actually begin to get a fairly full picture of how someone could develop, like you said, a range, not just one or two symptoms, but a range of symptoms based off this sort of core trend of coagulation. So I think it's cool to be able to pull some of those things together. So yeah. Well, then, do you have any last thoughts? I basically had to let you go to keep doing work. It makes me laugh. I'm like, just go back to the lab, run more samples. But yeah, I guess then what are your, last thoughts to leave off on?
I think the last thoughts would be to the patients, both long COVID and ME/CFS patients. I really would just want to tell them, please don't give up. I know it's easy for me to say with not having long COVID, not having ME/CFS. It's easy for me to tell you not to give up, but please don't, because we are really a team of dedicated individuals, and we are working really long hours. We don't take breaks. We work over weekends. We work late into the night. We get samples, not asking, for these samples. We just get the samples, and we are really working hard. So please don't give up. I think we're onto something. I hope our research would give help to and give some insights for treatment in future. So just keep on following us and thinking of us, and thank you for all the support. But most importantly, don't give up.
I agree. Yeah, thank you for that, Resia. Yes. Okay. Well, then thank you so much for talking today. And your work is incredibly interesting. I feel like we should check back in couple months from now, and I'm sure you'll have a million updates for me, which would be amazing. So maybe we can do that. But thank you for coming on today.
Thank you very much, Amy.
Okay.